So here we'll look at the events that occur when a neuron is stimulated, that is when it responds to a stimulus in the environment. All right, so usually what happens is that a stimulus in the environment is received by the dendrites and the soma of our neuron. All right, so the dendrites and the soma respond to any kind of stimuli. So we'll talk about stimuli in the next slide here, what kind of stimulus can get a neuron to respond. But regardless of what the stimulus is, the dendrites and the soma respond by creating a very localized, that is a very non-widespread electrical change called a graded potential. Okay. If this graded potential is strong enough, the axon will create a long-distance electrical signal that spreads or propagates down it, called the action potential. Right. So, and the action potential always proceeds from the axon hillock here down the length of the axon, as indicated by these gold arrows that you see here, toward our axon terminals. All right. The electrical signal of the action potential results in the release of a chemical signal that we call the neurotransmitter from the ends of our axon there. And so this neurotransmitter is what signals our next cell in the chain here. So if you think of two neurons kind of chained together, one neuron making a connection or a synapse with another neuron, this chemical signal, this neurotransmitter, will serve to signal the next cell in the line there, that next neuron, if it happens to be a neuron. All right. So the types of stimuli that neurons are responsive to, or the types of stimuli that sensory receptors or special sensory structures deliver to our neurons. Our main focus for Chapter 11 will be chemical stimuli or chemical signals. Particularly the chemical signal that we're going to spend the most time focusing on is a neurotransmitter. So when you think of the nervous system is really just a bunch of neurons kind of chained together end to end. And so neurotransmitters, which are released by neurons, serve as chemical stimuli for the next neuron in the chain there. So this is the kind of signaling that we'll focus on for this chapter here. Things like odors are actually airborne chemicals, airborne stimuli. Taste is simply a chemical that's dissolved in a liquid or a fluid like water or saliva. Chemical signals can also be released from damaged cells and damaged tissues that get our nervous system to respond and rally the immune system and things like that. In Chapter 13, we'll take a look at a couple of other stimuli that caused neurons to respond. These other stimuli are things like temperature, that is hot and cold on our skin. Mechanical stimuli, and these are things like touch and pressure. Mechanical stimuli are stimuli of motion and movement. Right? And other stimuli that are not chemical in nature, like light that is received by special sensory structures in our eyes and conveyed to our central nervous system. As I mentioned, our main focus here will be neurotransmitters as stimuli. Right? So our general overview of the nervous system signaling will begin with looking at connections from one neuron to another neuron, or neuron-neuron connections. So we have to introduce a couple of new terms here, a couple of old terms here, just to kind of refresh. So in a neuron-neuron connection, or I should say, when a neuron makes a connection with another cell, we call that a synapse. Right? So this is simply a connection between a neuron and some other cell, where the axon terminals of the neuron in question will make connection with that other cell. In Chapter 9, we saw that the synapse had a special name. It was called the neuromuscular junction. But basically what we saw was our axon terminals making a physical connection with our skeletal muscle cells. Right? This synapse always occurs across a fluid-filled gap. So that is, the neuron doesn't physically touch or physically connect to the next cell in the line in the chain there, and that there's always this fluid-filled gap in between them that we refer to as the synaptic cleft. And the reason for this fluid-filled gap is, recall that the neurons signal other cells by releasing a chemical signal. They release a neurotransmitter. So we have to have a fluid-filled gap in order for that chemical to be able to cross to reach our next cell. Right? couple of new terms here. So we've seen synaptic cleft before, so hopefully that's nothing new there. One new term that we'll kind of introduce ourselves to here is that if we think about two neurons in a chain, right, so very simple chain of neurons, this will be neuron 1, this will be neuron 2 here. The neuron that's releasing our neurotransmitter is going to be referred to here as our presynaptic cell. So presynaptic means before the synapse. So this is our neuron that's going to be releasing our neurotransmitter. And then receiving that neurotransmitter and responding to it is what we call our postsynaptic cell. So it's going to be receiving and responding to the neurotransmitter that was released by our presynaptic cell. All right. So we've discussed neuron-neuron connection. So a couple of new terms there, a couple of revisited terms. In also thinking about neuron signaling, we've also got to revisit really briefly the resting membrane voltage or resting membrane potential. Because what neurons are going to do when they receive a stimulus is that they're going to undergo a brief change in their membrane voltage or membrane potential. So let's revisit that for a second, just kind of review a few terms here. So recall that we call a cell that is not receiving a stimulus, that is a cell at rest, or we call that cell that's not receiving a stimulus a cell that is at rest. And at rest, all cells, neurons and muscle cells in particular are the ones we focused on, but all cells have a separation of electrical charge, or what we call a voltage across the plasma membrane. And we call this the resting membrane potential. Remember that this term potential here is synonymous with the term voltage. Right? Recall that this resting membrane potential, or this resting membrane voltage, let's say we have a little voltmeter here that we place across our plasma membrane. Our voltmeter will read an internal or intracellular voltage of minus 70 millivolts or minus 0.07 volts. So the inside of our plasma membrane is slightly negatively charged with respect to the extracellular or outside of our plasma membrane. Right? So most cells, including neurons, are going to read a voltage of minus 70 millivolts across the membrane. Does anybody recall what the resting membrane voltage was for a muscle cell? If you said minus 90 millivolts instead of minus 70 millivolts, you'd be correct. So had a slightly more negative charge with skeletal muscle cells than we see with nervous system cells. All right. So let's see. Also with the resting membrane voltage, recall that at rest on the outside of the cell and at rest on the inside of the cell, we saw two different concentrations or a concentration gradient of sodium and potassium. So recall that on the outside of the cell at rest, we saw that there were high concentrations of sodium and very low concentrations of potassium. Right, whereas it was the exact opposite on the intracellular environment. Low concentrations of sodium ions on the inside of our cell with high concentrations of potassium on the inside of the cell. Now, take a look here, and if you recall, both sodium and potassium are both positively charged. So again, you might be asking yourself, you might say, okay, with all these positive charges that are hanging around both inside and outside of our cell, how do we end up with a membrane voltage on the inside of the cell that's negatively charged? Right? And recall that this has to do with the ability of sodium and potassium to move in and out of the cell. And really the big determining factor in resting membrane voltage was how easy was it for potassium to leak out of the cell. Right? So recall that sodium and potassium can diffuse across the plasma membrane through what are called leak channels. So we have sodium leak channels and we have potassium leak channels. And you can kind of think of these as doorways that are always open. So through sodium leak channels, sodium is always going to diffuse from high concentration outside of the cell to the inside of the cell. Potassium leak channels, because potassium's at high concentration on the inside of the cell, low concentration outside at rest. Potassium's always going to leak or diffuse out. Right? So sodium diffuses into the cell, right, through these sodium leak channels. As I mentioned before, leak channels are a form of facilitated diffusion. That is, think of these as protein doorways through the membrane, because sodium and potassium can't diffuse directly through the membrane or directly across the plasma membrane. So because sodium is a positive charge and it's coming into our cell, it's going to make the inside of our cell more positive. Right? Potassium can leak out of our cell. Potassium also being a positive charge, with positive charge leaving the cell, that means our inside of our cell is going to become more negatively charged. Right? So with potassium leaving the cell, sodium entering the cell, how do we still end up with the negative voltage that is our resting membrane voltage? And recall that potassium can leave the cell much more easily than sodium can ever enter the cell. And why is that? Really, it simply comes down to the fact that there are more potassium leak channels in the plasma membrane than there are sodium leak channels. So there's simply more doorways for potassium to get out of the cell than there is for sodium to enter the cell. All right. So potassium leak channels are much more abundant than sodium leak channels, which, remember, the PhysioX lab that we did in chapter 3, that was PhysioX lab 1. We saw that when we increased the number of plasma membrane proteins in facilitated diffusion, we get a faster rate. So really, all we're adding is more plasma membrane proteins that allow potassium to cross. So it's much easier and much faster for potassium to diffuse and leave the cell than it is for sodium to get into and enter the cell. Right? This, to give you kind of a figure or number here, there are somewhere around 25 times more potassium leak channels than sodium leak channels in the membrane. So it's really easy for potassium to leave the cell, and really difficult for sodium to get into the cell. Right? So because it's really easy for positively charged potassium to leave the cell, this makes our membrane voltage much more negative on the inside of the cell than on the outside. All right, so it's much easier for potassium, a positive charge, to leave the cell than it is for sodium to enter the cell. And recall also that these leak channels, if we didn't have something to keep the concentrations of sodium and potassium at their high levels on the inside or outside of the cell, we wouldn't get this leakage. But we have a structure, we have a membrane protein that's called the sodium-potassium pump, or the NAK pump, that compensates for the fact that diffusion of sodium and potassium is occurring across the membrane. Right? So our sodium-potassium pump uses 1 ATP. Because sodium is leaking into the cell, it's going to kick 3 sodium out for that 1 ATP. Potassium is going to leak out of the cell, and so it's going to bring some potassium back in. So for each ATP that it spends, it brings into potassium. So we're kicking out 3 sodium that have diffused or leaked in, and kicking out or bringing in 2 potassium that have leaked out. Also recall, too, that the cell has a number of large negatively charged proteins inside of the cell that can't cross the plasma membrane. These contribute a little bit of that negative charge, or to our negative resting membrane voltage, but they're not a huge contributor of it, simply because of the fact that potassium is, as a positive charge, is really easy for potassium to leave the cell through those abundant potassium leak channels. So the fact that potassium leaves and takes positive charge with it has a much greater effect on creating the resting membrane voltage than these large negatively charged proteins do. All right, and what I put here is a diagram in your PowerPoint presentation, so you can review all of the individual things that are going on during the resting membrane voltage. So be sure to pull that up, take a look at it, make sure you're comfortable with that, those concepts and those ideas here before you move on in the nervous system. All right, so we'll move past that, and we'll take a look at the start of neuron signaling. So we'll start looking at the graded potentials that are created when a neuron receives a stimulus. Right. So we talked a little bit earlier about how there are many different stimuli that cause neurons to respond, but our focus here is going to be a neuron here. So this is an axon terminal of a presynaptic neuron, releasing neurotransmitter here, this sort of greenish blue substance here, and that neurotransmitter is making contact with the soma and the cell body and the dendrites of a postsynaptic neuron or a neuron receiving our neurotransmitter. All right, so this is what we're going to focus on here. So from here, what we're going to assume is a neurotransmitter has been released and our postsynaptic cell is responding. So we're going to look at how the postsynaptic cell that's receiving our neurotransmitter is going to respond to that. All right. So we've got our neurotransmitter, our stimulus, that has been released from our presynaptic cell, and our postsynaptic cell here will respond to it. And it responds by creating an electrical signal called a graded potential. Right? Recall that the term potential means voltage. So a graded potential, just like an action potential, and we had really two types of electrical signals. We had graded potential and action potential. And both of those potentials are changes in membrane voltage. But in this case, the graded potential, it only occurs in very specific structures of the neuron. It occurs in the dendrites of the neuron or in the soma of the neuron, but never in the axon. The axon is action potential territory. So it's kind of easy to think of A as an axon. Axons create action potentials, right? Dendrites and somas create graded potentials. So what is a graded potential? The strength of a graded potential is proportional to the stimulus strength. So that's why they're called graded, because it depends on how strong our particular stimulus was. In this case, our stimulus was a neurotransmitter. So this would mean that if there is more neurotransmitter being released from our presynaptic cell, that would result in a stronger graded potential or a stronger electrical change in our postsynaptic cell. All right. Graded potentials also have a feature where, graded potentials, they're not long distance signals, so they're very short local signals, or very short distance local signals. And the reason for that is that the strength of a graded potential decays with the distance from its origin or its point of origin. So if a graded potential, let's say, were to start right here on the tip of this little dendrite here, and let's say we were to measure it right here near the axon hillock, the strength of that particular graded potential that was created here would be much weaker if we measured it further away. If we get closer to it and measure it, it would be stronger. So the strength of the signal, the strength of the electrical signal, decays with distance, which is not something that we see in action potentials. Action potentials do not decay with distance from their origin point. Right? So we'll take a look at what happens during a graded potential really briefly. All right, so we get a little section of our plasma membrane here. Recall that neurotransmitters have to bind to a neurotransmitter receptor. In skeletal muscle cells, we saw these as ACH receptors. But here we'll just kind of go with a general neurotransmitter here. And we've also got some sodium on the outside of a cell, our cell here. And our cell is at resting membrane voltage because it's at minus 70 millivolts here, as indicated by our little voltmeter across the plasma membrane. Right? So our neuron is currently at rest. No stimulus is being applied yet, anyway. But it will be. So we've got some neurotransmitter that's moving toward our neurotransmitter receptor. Okay. So this neurotransmitter in this case is going to bind to a protein receptor on our postsynaptic cell. And this receptor is usually called a chemically gated channel, in that a neurotransmitter, a chemical, is what's going to open this particular channel when the neurotransmitter binds to it. All right, so like I said, this receptor is actually a channel, and we call it chemically gated because if you think of the channel as a closed door, the neurotransmitter, which is a chemical, is going to be our key to opening that door. All right. So the reason that we only see graded potentials being created in the dendrites and in the soma of our neurons is because these chemically gated channels are only found in the dendrites and in the soma. They're not found in the axon terminal. Okay, so the neuron is only going to be able to respond to neurotransmitter if that neurotransmitter is in the proximity of its dendrites or its soma. So if neurotransmitter doesn't bind to our neurotransmitter receptor or chemically gated channel here, the cell is not going to respond to it. Okay. When the neurotransmitter binds, it's going to open this chemically gated channel. In our example here, our chemically gated channel or neurotransmitter receptor is going to let sodium into our cell here. Okay. So what we're seeing here, what we'll see here, is that this channel will open. Sodium is going to come into our cell, in this case, and our membrane voltage is going to change a little bit because sodium is a positively charged ion. So when it comes into our cell, that's going to change our resting. That's going to change our cell from resting membrane voltage to, in this case, to a more positive voltage, a slightly more positive voltage at minus 20 millivolts. This number is just completely arbitrary. I was just giving it as an example here. Okay, but this is not this is a realistic number that that a graded potential might create. All right. In this case, sodium entered the cell, but we could also have exit of substances through chemically gated channels. And then these entry or exit of substances would cause voltage changes in this little part of the plasma membrane here. Okay. So chemically gated channels, neurotransmitter receptors, are channels that that substances will move through by facilitated diffusion. So remember, facilitated diffusion requires a doorway through the plasma membrane. So in this case, because it's diffusion, it's still diffusion, we're going to get substances moving from a region of high concentration, right, to a region of low concentration, right? And recall that diffusion is always a form of passive transport. So I'll ask here, do you recall whether ATP was required for passive transport? If you said no to that answer, ATP is not required for passive transport, that's correct. That is a form of passive transport. Okay, so we see what happened. We saw what happened when a neurotransmitter binds to a chemically gated channel or receptor. We described the graded potential as a signal, an electrical signal that decays with distance, and so we'll see what happens. We'll see what we mean by that, and that these graded potentials are always local electrical signals, and that local implies that the signal strength is going to get weaker as we move away from its point of origin here. So to illustrate this particular example here, we've got a little section of a plasma membrane. We've got some distance between our chemically gated channel here and a part of the membrane down here. Okay. And we'll assume that this neurotransmitter opened up a chemically gated channel that allowed sodium to diffuse into our cell. So that's what this purple is. This is our sodium that is diffused into our cell. And if we were to take a measurement, right, if we were to put a little voltmeter into the plasma membrane near our chemically gated channel receptor, we would find that the membrane voltage is actually slightly more positively charged, right? So sodium comes in, brings positive charge with it. And we see that our membrane voltage is about plus 50 millivolts above resting membrane voltage. So it went from minus 70 to minus 20 millivolts. So it got closer to zero here, right? And that was measuring our membrane voltage right next to our chemically gated channel here. But let's say we move a little bit further down and we put a voltage meter a little bit further away from where our signal originated, and we'll measure somewhere around maybe minus 50 millivolts. So still slightly positively charged, but not as much, right? So we'll move a little further away. Our voltage is now only plus 20 millivolts above resting membrane voltage. So that means that our signal has decayed a little bit. That is, it got a little bit weaker as we moved away from where it started. Right? And if let's say one just final example, just to round out our example here, we'll put a voltage meter way over here, and we'll see that our voltage meter maybe doesn't read anything different than resting membrane voltage. So we're still at minus 70 millivolts all the way down here. Right? So our signal got weaker as we got further away from where it started. Right? Okay, so I gave an example where a neurotransmitter opens a channel that lets sodium into the cell, but this isn't always going to be the case. Sodium is not always going to come into the cell when a neurotransmitter opens a channel. Some graded potentials make the membrane more positive in that the chemically gated channel that opens might let a substance like sodium into the cell where it's going to bring positive charge. Right. So in this case, a stimulus that makes the membrane voltage more positive for a brief time or moves the membrane voltage closer to zero here is called a depolarizing stimulus. So in this graph, in this example here, we have membrane voltage on the vertical axis here with minus 70 being our resting membrane voltage before the stimulus was received. And we've got time in milliseconds on the horizontal axis here. We'll release a neurotransmitter or we'll apply a stimulus that depolarizes our cell. So we'll create a depolarizing stimulus, and then the cell will respond by opening channels that let positive charge into the cell. All right. So depolarizations, if it's a neurotransmitter that's opening chemically gated channels, depolarization would be caused by sodium entering through these voltage gated, these chemically gated channels here that were opened by our neurotransmitter. Okay, so this would be a depolarizing stimulus if sodium were to enter our cell. There are two neurotransmitters in particular that I just want you to be familiar with that cause depolarizing or create a depolarization in the cell. One of these neurotransmitters we've seen before, it's called acetylcholine or ACH. So recall that when our neuron released acetylcholine and signaled our skeletal muscle cell, that our skeletal muscle cell responded by letting sodium into it, bringing positive charge into our cell and making the membrane slightly more positively charged where the sodium entered that part of the cell. Right. There's also another neurotransmitter called norepinephrine or NE that creates that tends to create depolarizations in neurons that receive this neurotransmitter. So norepinephrine will also open chemically gated sodium channels in the plasma membrane. Right? But not all stimuli make the membrane voltage more positive. Some of them actually make the membrane voltage more negative for a brief time. All right. So again, we have our same setup here, membrane voltage on the vertical axis, time on the horizontal axis here in milliseconds, so thousandths of a second, 1/1000th of a second, 2/1000th of a second. So these are very brief periods in time. And again, we have our membrane voltage being recorded by our voltmeter across the membrane. So we've got resting membrane voltage here. A stimulus will be applied here that makes, in this sort of dark blue region here, that makes the membrane voltage more negative for a brief time. So the term that we use when a stimulus makes the membrane voltage more negative for a brief time is what we call a hyperpolarizing stimulus, right? Or if it makes our membrane voltage move further from zero, right? So it becomes more negative, right? Hyperpolarizing stimuli are, in this case of neurotransmitters, our stimulus is opening a chemically gated potassium channel. So it's letting potassium leave the cell. Recall that potassium is a positive charge, and it's at high concentrations on the inside of our cell and rest. So if it's allowed to leave the cell, it's going to take positive charge with it, and our cell is going to become more negatively charged for a brief time. Right? I'll leave you with one example here of a neurotransmitter that creates a hyperpolarizing stimulus. That's a neurotransmitter called gamma-aminobutyric acid, or GABA for short. This is a common hyperpolarizing neurotransmitter in the brain. It's hyperpolarizing are also referred to as inhibitory neurotransmitters for a reason that will become apparent when we look at the action potential, which will be in the next segment.